Welcome to all. Myself Sajithra, lecturer of Emmanuel Arthur College of Pharmacy. In this session, we completely discuss about carbohydrate metabolism. First of all, what is carbohydrates? Carbohydrates are most abundant organic molecule on earth. Carbohydrates are defined as aldehyde or ketone derivatives of polyhydric alcohol. For example, glycerol on oxidation, which is converted to diglyceraldehyde, which is a carbohydrate derived from the trihydric alcohol glycerol. And all the carbohydrates have the general formula, which is CnH2nOn. Then carbohydrates are classified into four main categories. This classification was based on the sugar moieties present in a compound. First of all, monosaccharides, the carbohydrates which contains only one sugar moiety, which is known as monosaccharides. If the carbohydrate which contains two sugar moiety, it is known as disaccharide. And the carbohydrates which contains more than 2 and less than 10 carbohydrates, which is known as oligosaccharides. And the carbohydrates which contains more than 10 sugar moiety, which is known as polysaccharides. Functions of carbohydrates. Main source of energy in the body. Energy production from carbohydrates will be 4k calories per gram. Storage form of energy. Excess carbohydrate is converted to fat. Glycoprotein and glycolipids are components of cell membranes and receptors. Structural basis of many organisms. Example, cellulose of plant, exoskeleton of insects, etc. Biomedical importance of glucose. Glucose is a major carbohydrate. It is a major fuel of tissues. It is converted into other carbohydrates in the glycogen for storage, ribose in nucleic acid, galactose in lactose of milk. They form glycoprotein and proteoglycans. They present in some lipoproteins also. This glucose may be present in plasma membrane in the form of glycohalax. And then we see about the metabolism. Thousands of chemical reactions are taking place inside a cell in a organized, well coordinated and purposeful manner. All these reactions are called as metabolism. Types of metabolic pathway. The metabolic pathway are classified into three main classes. They are catabolic pathway, anabolic pathway, and ambifolic pathway. And stages and phases of metabolism. There are three stages. They are primary stage, secondary stage, and tertiary stage. Then we see about the major carbohydrate pathways metabolism. First of all, carbohydrates metabolism means biochemical process involved in synthesis, breakdown, and interconversion of carbohydrates in living organisms are collectively called as carbohydrate metabolism. Glucose is the essential molecule of carbohydrate metabolism which participates in various metabolic pathways. Insulin is the primary metabolic hormone synthesized in pancreas and regulates glucose level in blood. During photosynthesis, plants utilize atmospheric gases to manufacture carbohydrates and store it as the starch or lipids. Animals and fungi consume plant contents for energy production. 1 gram of carbohydrates and lipid oxidation gives approximately 4 kilocalorie and 9 kcal of energy, respectively. In aerobic organisms, glucose is metabolized in the presence of oxygen to yield carbon dioxide, water, and energy in the form of ATP. Carbohydrate metabolism is simpler than that of fat and amino acids. Therefore, carbohydrate is utilized as immediate source of energy. In animals, glucose is the primary source of energy. In some other organisms, glucose polymers are broken down during digestion and are absorbed as glucose. There are various carbohydrate metabolism pathway take place in our body. They are glycolysis, citric acid cycle, gluconeogenesis, glyconeolysis, hexose monophosphate stand, uronic acid pathway, galactose metabolism, fructose metabolism, amino sugar metabolism. Entry of glucose into the cell. Insulin dependent transport system of glucose, non dependent hormone insulin. This is operative in hepatocytes, erythrocytes, and brain. Insulin dependent transport, that is muscles and adipose tissue. Okay, then discuss about the first important metabolic pathway, that is glycolysis. Glycolysis is also known as EMP pathway, and which is defined as the glycolysis is defined as the sequence of reactions converting glucose to pyruvate or lactate with the production of ATP. Features of this glycolysis are it takes place in all cells of the body, enzyme present in cytosol fraction of the cells. Lactate is the end product in the anaerobic condition, and pyruvate is the end product of aerobic conditions. Tissues lacking mitochondria, which is the major pathway for the ATP synthesis. Then reaction of glycolysis. The first reaction, which is energy investment phase or primary phase, second one splitting phase, and third one energy generating phase. This is the pathway. First we see about the energy investment phase. It starts from the glucose 2 and it ends from the production of fructose 6 phosphate. First, in this energy investment phase, glucose molecule is phosphorylated to glucose 6 phosphate. ATP splits into ADP and phosphate group in the presence of enzyme hexokinase. The inorganic phosphate performs phosphorylation of glucose to yield glucose 6 phosphate. Energy produced by the hydrolysis of ATP is used for forward reactions. This reaction is irreversible. Glucose 6 phosphate undergo isomerization to yield fructose 6 phosphate under the influence of enzyme phosphohexose isomerase. Fructose 6 phosphate undergo phosphorylation to yield fructose 1,6 bisphosphate under the influence of enzyme phosphofructokinase. It is an irreversible reaction and utilizes one molecule of ATP. Then we see about the splitting phase. This phase includes the following reaction, that is this fructose 1,6 bisphosphate cleaves into two compounds, each having three carbon atoms, namely glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Under the influence of aldolase enzyme, dihydroxyacetone phosphate undergoes isomerization to yield glyceraldehyde 3 phosphate under the influence of enzyme phosphotriose isomerase. Then this glyceraldehyde 3 phosphate is dehydrogenated and concurrently phosphorylated to 1,3 bisphosphoglycerate with the help of NAD+. The enzyme used in this step is glyceraldehyde 3 phosphate dehydrogenase. Then see about the energy generating phase, and this energy generating phase is the third phase of this glycolysis. One glucose molecule yield 4 ATP molecules in two steps. 3 bisphosphoglycerate yield 3 phosphoglycerate under the influence of enzyme phosphoglycerate kinase. This reaction is an example of substrate level phosphorylation which involves formation of 1 ATP molecules. Then 3-phosphoglycerate isomerase to yield 2-phosphoglycerate in the presence of enzyme phosphoglucomutase. This reaction involves shifting of the phosphate group from third to second carbon. Then 2-phosphoglycerate forms phosphophenol pyruvate in the presence of enzyme enolase. In this step, one water molecule is removed and high energy phosphate bond is formed. The enolase enzyme requires magnesium and its activity is inhibited by fluoride. Phosphoenol pyruvate undergo dephosphorylation reaction to yield pyruvate by enzyme pyruvate kinase. It is an irreversible reaction and generates 1 ATP molecules. Under anaerobic condition, and pyruvate is reduced to lactate in the presence of enzyme lactate dehydrogenase. This lactate then enters the cycles, while under aerobic condition, pyruvate enters the citric acid cycle for complete oxidation. Then see about the energy. One cycle utilizes totally 8 ATP. That is, first it in glyceraldehyde 3 phosphate dehydrogenases yield 6 ATP and then phosphoglycerate kinase yield 2 energy and pyruvate kinase yield 2 energy and finally phosphofructokinase and hexokinase utilizes 2 energy and it is minused by the synthesized energy. So, the net ATP synthesized in glycolysis under aerobic condition is 8 and it synthesized 6 pyruvate at the anaerobic condition. And then significance of this glycolysis is it is an important due to the following reasons. Energy is obtained in the form of ATP, which is used further for various metabolic processes. Glyceraldehyde 3 phosphate is utilized in triglycerides and phospholipid synthesis. Pyruvate is used in the biosynthesis of amino acids alanine by transamination. And then acetyl-CoA is essential for biosynthesis of cholesterol. In RBC's glycolysis pathway is the only source of energy. During excessive physical exercise, body muscles lack sufficient oxygen. Therefore, anaerobic glycolysis occurs in muscles to fulfill energy requirements of body. Glycolysis is the first step of complete oxidation of glucose. The glycolytic cycle provides various biochemical compounds which are used for the synthesis of non-essential amino acids and fats. These are the significance of glycolysis and this and there is an outline for now see the overview of this glycolysis. Glycolysis in aerobic condition it yield pyruvate and anaerobic condition it yield the lactate. And I hope you are able to understand them. You have able to understand this session. Thank you for watching.